Okay, let's go on and talk about what goes on inside the cell membrane and the rest of cell structure. Now, when you think of what I'm going to ask you on a test, this probably is where the bulk of the information for exam 2 tests on the cell is going to come from. I really do want you to know the cell's structures, the internal organelles, and their functions. So as I go through these, I'll point things out. You may want to do some flashcards. You may want to do some extra notes just to make sure you keep track of everything that's going on inside the cell. We really do need to know these little bits and pieces that you find inside a cell. So we'll start off by saying on the outside of a cell is a membrane, and on the inside is the cytoplasm, and that's where I ended the previous lecture. Talking about the cytosol as being the area inside the membrane. So let's start looking at what's inside the membrane. So we can start off by saying the things that are non-membranous structures, we can start off with those and then kind of look at the organelles with single membrane and then with double membranes, and it's a way to organize things. So you need to know that ribosomes are found in all cells that are biologically active. So ribosomes are going to be found in all cells. And the second bullet point, ribosomes are the site of protein synthesis and translation. Okay, ribosomes are the sites of protein synthesis and translation. That's the function of ribosomes. For all of these organelles, you're going to look for structure and function. Okay, so their function is they're the site of protein synthesis. Now their structure, we can say that they're not surrounded by a membrane, but they're composed of two subunits, a small and large subunit, and those subunits are made up of protein and RNA. Okay, prokaryotic ribosomes and eukaryotic ribosomes are slightly different. And also, it's kind of interesting to know that there's actually organelles that contain their very own ribosomes. So again, this is our first not membrane-bound structure that you need to know inside the cell membrane. The next is the cytoskeleton. This is showing you here a cytoskeleton within a cell, a variety of sort of wires and tubes that make up the cytoskeleton. So it's fair to say that the cytoskeleton is not just one thing, but it's a little bit more complex. It's a dynamic network of structural proteins, and the cytoskeleton is made up of proteins. It's a key element. It's made up of proteins. It can be organized and reorganized into different shapes. And here's our function. It helps to maintain the cell's shape. It's also important in division and movement. Okay, so let's look at those structures that make up the cytoskeleton. The thinnest of them is called a microfilament. Okay, the microfilaments include actin. Actin is a monomer or a single, it's made up of single subunits that are pushed together. It can get longer or shorter in helping to assist the cell's movement, and it's also really important in terms of maintaining a cell shape. Microfilaments are important in terms of dividing the cytoplasm during cell division. Microtubules are thicker and they look like tubes. If you were to describe them, you would make a straw to describe them. These are the thickest of the elements, and they're composed of tubulin monomers. So tubulin is a little a round or globular structure that can be joined together to form these tubes. And what's really interesting is the cell has the ability to make these tubulin or the microtubules longer and shorter. There's specialized areas within the cell that we call microtubule organizing centers that are the places that lengthen and shorten these tubes. These are really important for moving things because as they get longer, they can push things away. As they get shorter, they can pull things closer towards them. And they help to move the organelles and chromosomes around within a cell. So when we talk about cell divisions where you divide chromosomes, it's microtubules that actually pull those chromosomes apart. Here's a picture of intermediate filaments, and for your purpose, just understand that these are, again, fibers that are somewhat in between the actin and the tubulin or the microtubule fibers, and they have a variety of different thicknesses, have a variety of different functions. They're relatively stable, and they help to keep the cell from stretching too completely. Within the cytoskeleton, we're also going to find motor proteins. These attach to our organelles sometimes. They attach to the cytoskeleton, and they move things along the cytoskeleton. So you can see this Dynin is a motor protein that can move along, can attach to an actin and move along an actin fiber. These things assist in cell movement. When we talk about cilia and flagella, which are made of those cytoskeletal elements, these are the things that actually move things along. And some of the motor proteins include the kinesins and dynins. These are what interact with the microtubules. The myosins are what interact with the microfilaments. Okay, so let's go ahead and talk about some elements that are made up of microtubules. Cytoskeletal elements that extend outside of the body of the cell include flagella and cilia. Flagella are generally considered longer, and there are fewer of them, although in some bacteria you're going to find some long flagella where there's multiple flagella. Cilia, again, are shorter, and they tend to be far more of those. So what they do is they tend to move things. They're made up of microtubules that are arranged in what we call a 9-in-2 arrangement, with 9 double microtubules around the outside and 2 single pairs in the middle. And motor proteins and microfilaments hold these structures together. These are attached to the base they're attached at the base to something called a basal body, which is inside the cell and holds these elements in place. So here's a really neat picture of the internal structure of cilia and flagella. And again, these things have the ability to move back and forth in a sort of sweeping motion. Okay? Again, here's a picture of the movement of a cilia, again, sort of moving back and forward. Now again, we usually refer to cilia, and you can see some cilia here on the surface of a paramecium. Here's some respiratory cilia as working in like a sweeping fashion. They sweep things past them. In human bodies, cilia are found in places where you want to sweep things in one direction or another. The respiratory epithelia have a really interesting function for the cilia. They actually, when you breathe things in, there's mucus that sticks to the cilia, and the mucus catches particles that get breathed into your respiratory tract, and these cilia sweep them back up into the back of your throat, and then hopefully you'll swallow whatever dirt or bacteria make their way into your respiratory tract before they make it further into your respiratory tract and cause infection. So these are really important in terms of keeping you from getting sick. And I can prove it because one of the interesting facts about these respiratory epithelia is cigarette smoking will actually kill the respiratory epithelia, and without them, we know that people who smoke cigarettes tend to have much more in terms of lower respiratory tract infections, which is pneumonia, and also bronchitis, than folks who have functioning respiratory epithelia. Now the cool thing with this is these respiratory epithelia actually replace themselves every couple weeks. So for those of you who are smoking, one of the beautiful things about quitting smoking is usually within about 2 or 3 weeks of quitting smoking, you'll notice that these cells begin working again, and you'll notice that you have less upper respiratory tract infections. So it's a really good reason to remind your friends who smoke that it's a good thing to quit smoking. Take care of their respiratory epithelia. Take care of their respiratory cilia. Okay. The only human cell that has a flagella is a sperm cell, and again, it's important in terms of movement. Again, you can see the picture of the flagella, much longer than the cilia, and again, the only thing that you need to know for your purposes is the flagella are the unique human cell that has a flagellum. The microtubule organizing centers include those basal bodies. Remember, we said that they held the cilia and flagella in place, and also something called centrioles. These centrioles are found near the nucleus of eukaryotic cells, are important in cell division. They actually help to organize the microtubules that separate our chromosomes during cell division. So know that centrioles are found in pairs. They're microtubule organizing centers found in pairs near the nucleus, and their function is to organize the microtubules that separate our chromosomes during cell division. Know that basal bodies are the base of cilia and flagella, and they help to build and organize and control the cilia and flagella. Okay, so again, we've made it through our organelles that don't really have a membrane that surrounds them. Now it's time to start talking about the organelles that are surrounded by a single membrane. So we can start talking about something called the cytomembrane system. We're going to describe different structures, But as you can see in this picture, things tend to flow from one structure to the next. So I'll talk first about the rough endoplasmic reticulum and then the smooth endoplasmic reticulum, but you can see that they're somewhat tied together. So we can talk about an overall ER, but we can also talk about things that break off from here that are also part of this cytomembrane system. Okay, so the ER, or endoplasmic reticulum, and you won't have to spell this, you'll just have to remember the letters ER. It's a series of fluid-filled tubes that are continuous with the outside of the nuclear envelope, and they're a passageway for things to move or to travel through the cells. They're also a place where things are made. Okay, we can describe again a rough and a smooth endoplasmic reticulum. You can see them in this picture as looking rough and smooth. These little dots are what make the rough endoplasmic reticulum rough, and those dots are actually ribosomes. If you remember from the earlier discussion, ribosomes are the site of protein synthesis. So if I ask you what the rough endoplasmic reticulum does, and you know that the ribosomes are there, you can say, okay, well this is where we can make and on the ribosomes and then transport proteins that are newly made inside the cytoplasm. Okay, again, they also carry out a variety of modifications to proteins. They add carbohydrates, and these carbohydrates are really important when you add them onto proteins because they function as a system by which the cell guides proteins as they move from one place to the next. Here's a picture of the smooth ER. Again, a branch-like system of fluid-filled tubes. Again, there's a variety of enzymes in here, and they're important in modifying some of the proteins that are made in the rough ER. These are also important for synthesizing and assembling lipids, and also for storing molecules. Now, pieces of the smooth endoplasmic reticulum can kind of bud off, or they can break off. You can imagine this one here, just sort of the membrane surrounding itself and breaking off, and the contents can be broken off and can give rise to a membrane-bound structure that can travel from one place to another. We call that a vesicle. Okay, so the vesicle is the next part of our endomembrane system. A vesicle can be considered a vehicle for transporting materials through the cell. That's a good function, so make sure you think of the vesicles as being used for transport, also for storage. They're formed by budding off or breaking off of a part of the ER, and then also you'll see them being formed by breaking off of another organelle called the Golgi. Also, when the cell folds inwards and surrounds something, it forms vesicles by a process called endocytosis. So the vesicles can travel along the cytoskeleton, they'll move along the cytoskeleton, and they can actually fuse or deliver their material into another organelle, and the membrane will fuse together with the other membrane and dump its contents inside. The other thing that happens with the vesicles is some of them become specialized and they actually carry out separate functions. They're not just for transport or storage anymore. So here's a beautiful picture of an endosome, and this picture here shows, this is the outside of a cell, this is the cytoplasm. What's going on is some of the contents outside the membrane are surrounded by the cell membrane. It folds inwards, surrounds them, and takes them into the cell with an endosome. And again, this is an example of a specific vesicle made by the process of endocytosis. Taking something in from outside the cell is called endocytosis. If it's something solid, we call it phagocytosis. If it's something liquid, we call it pinocytosis. You can use endocytosis, phagocytosis, and pinocytosis somewhat synonymously, so they're somewhat very similar terms. Okay. Now here's another an example of a specialized vesicle. This is, again, one of my favorite pictures is a picture of a developing human embryo. If I were to look at you at 48 days of development, I would note that you didn't have any specialized areas where your fingers were developing, but you can see there's some areas of what's going to become your hand where cells have to be destroyed in order to make your future hand. There's actually a process by which the cell breaks down cells, by which the organism breaks down cells that it doesn't need. Cells are programmed to die. Okay, these cells here that we don't need anymore are programmed to die, and that idea of killing themselves to perfect the total organism is a sort of programmed cell death called apoptosis. Okay, the cells that help these cells to break down are called lysosomes. Lysosomes are vesicles that are filled with catabolic enzymes. These are enzymes that can digest all sorts of things, They can digest basically any of the molecules that make up cells, but as long as these enzymes are within the cell, they don't cause any damage. As soon as they're fused with another membrane, they can digest whatever's inside that membrane. In this case, when we break open the cell membrane and those enzymes are released, it actually causes the death of cells, and that's the apoptosis part of what the lysosomes do. Lysosomes are probably my favorite organelle, so make sure you take a little time and look at those and make sure you understand that they're vesicles with a specialized function of cell digestion, and anticipate probably seeing that on future assessments. Okay, peroxisomes are another specialized organelle. They actually contain oxidative enzymes. In particular, peroxisomes contain catalase, which is an enzyme that breaks down peroxide. You may be familiar with peroxide. You may have poured it on a cut when you were a little kid and you saw bubbling occur. That bubbling actually required enzymes that were within your cells, within these peroxisomes. We actually, within our cells, can make peroxide. We don't make a lot of it, but it is a byproduct of metabolism, and by breaking it down, that actually protects our cells. So it's an important component of maintaining the integrity of your cells. Find a lot of those peroxisomes in your kidneys and other metabolically active organs. Here's a picture of a specialized vesicle that you find within plants. It's called a vacuole. And vacuoles, for the most part, are large in plants and protozoans, and they're used for storage. One of the important things that they do is they store water. Just recently, my husband bought me roses, and I forgot to put the roses in water. And so what happened is the rose stems, which are normally more rigid, and the rose looks all beautiful, the rose stems remain rigid because these vacuoles are filled with water. When the water evaporates, there's nothing pushing against the cell wall on the outside, and the flowers wilt. A good reminder that you also always should water your flowers to maintain turgor pressure, or the pressure of the water against the walls inside the cells, in order to keep your flowers from wilting. Okay, here's a neat picture of another membrane of the another element in the endomembrane system called the Golgi body. The Golgi body is a series of membranes that are sort of stacked together like a stack of pancakes. They can interact. Materials from one membrane can be passed along to the next membrane, and they're a place where materials that are made in the ER or made in other places can be packaged and transported and prepared for exocytosis. So things that are made in here can be packaged, and then they can be prepared to be sent outside the cell for the process of exocytosis. An example that I always like to give with exocytosis is talking about the case of neurotransmitters. In order for you to hear my voice in your ears and that message to be transmitted from the cells in your ears to the cells in your brain that process the thoughts that you have, neurotransmitters send a message from one nerve cell to the next. In order for those neurotransmitters to be released, they have to be made within the cell, and they're released from one cell, in between one cell and the next, by a process of exocytosis. So you're constantly, you can imagine Golgi within your nerve cells, constantly preparing these neurotransmitters for the process of exocytosis. Okay, so we've been through that endomembrane system, through the one membrane organelles. Now we're going to move on to the double membrane organelles. Again, a test question I am likely to ask is, what are the three double membrane organelles? You need to know that they are the nucleus, the chloroplast, and the mitochondria. And they're really important organelles. Again, you can think about these as being really essential to most of life. Okay, so let's start with the nucleus. Again, two membranes surround it. Again, because the nucleus you've seen, because it's so key, I'm probably going to ask test questions on it. So spend some time learning about the nucleus in detail. It stores the genetic information. It is the site where your DNA is found. It's surrounded by what we call a nuclear envelope. The nuclear envelope has two double membrane layers, and then there's pores in those membranes. Okay, within the nucleus is something called the nucleolus. Okay, and then the nucleus itself contains this material we call nucleoplasm, which is filled with fluid and nucleotides that can make up the DNA and RNA that are found within. There's some proteins in there, some important enzymes. and then also the DNA, which is in the form of chromatin, is found in your nucleus. Okay, so here's a neat picture of the double layer membrane with the pores showing on the inside. Again, these pores are kind of neat. They're composed of 8 identical protein subunits that allow for the pore to form, and they regulate what goes into and outside the cell. Here's a kind of a cool picture of the nucleolus. Your key thing for the nucleolus, I just need you to know that it's a dense staining area. So if this is not staining as darkly, the nucleolus always shows up much more darkly when we stain the nucleus. And it is the site of ribosomal synthesis. This is where we make our ribosomes. Again, key element, I'm going to ask you what's the function of the nucleolus. You need to know this is the place where we make the subunits that make up the ribosomes. Okay, within the nucleus is the DNA. DNA in eukaryotic cells is linear. Okay, it's surrounded by proteins. The proteins are actually called histone proteins. They tend to be positively charged because DNA is negatively charged. The DNA surrounds or groups around the histone proteins, and it forms these things called nucleosomes. We'll find nucleosomes evenly spaced along the DNA with DNA wrapped around it. These nucleosomes can pack together, and they package very tightly to form chromosomes when we tighten up a long piece of DNA to make a nucleosome. Now, prokaryotic DNA is circular, and it doesn't have nucleosomes, so that's kind of a key thing. I need you to know that prokaryotic DNA is circular. It doesn't have those nucleosomes. Okay, so here's a nice picture of chromatin with the DNA wrapped around the histone proteins forming a nucleosome with the histone beads. And most of the time when you're in your cell, if I was to look at the nucleus and I could look at the DNA, it would look like this. This would be something I'd have to do under high magnification. But I'd see these nucleosomes, and I might see the string-like DNA. But just before your cells get ready to divide, this chromatin packages itself into chromosomes. Okay? Now again, this chromosome is literally one great big long piece of DNA all wound up very tightly here. Okay, this would be one piece of DNA, and on this other side would be its identical copy. So this X-shaped chromosome is 2 identical copies of the same DNA segment. Okay. Again, here's a nice picture of the nucleosome forming. Again, you can see these 2 identical copies of the chromosome tied together to form this duplicated chromosome. The place where they join together is called a centromere. The place where microtubules can join them is called the kinetochore. Again, here's DNA winding up to form a nucleosome, the nucleosome winding up to form chromosomes. Again, you can see chromosomes within the nucleus of a cell. Again, you're going to need to know a little bit about the anatomy of a chromosome. So let's start off by saying that you have 46 chromosomes, and they look pretty much like this picture here when they get ready to divide. Now, before they get ready to divide, they look more like this. Okay, but just prior to division, they wind up like this. And again, prior to division, they have to copy themselves. This would be one copy. This would be an identical copy of each of your chromosomes. So you'd have 2 identical copies. They join together at a place called the centromere. Okay, and I need you to know that term centromere. And know that these 2 identical copies are sometimes referred to as chromatids. You could call this chromatid 1, chromatid 2. Each of them has a complete set of complete information from one of your chromosomes. So each of these can make one of your chromosomes, but together they are a duplicated chromosome. Okay, let's go on and talk about, again, what do you need to know about your nucleus? Again, the nucleus is surrounded by a double membrane. So this is that structure. You need to know it has a nucleolus inside it. Again, that's structure. You need to know that it's filled with the nucleoplasm that also has chromatin in it, which can condense to form chromosomes. Okay, what's the function of DNA? The function of DNA, it's the storage, the function is storage of the genetic material, storage of genetic information, or the information for making proteins in the cell. The nucleolus, its function is to make ribosomes. Okay, let's go on and talk about the mitochondria now. Sorry for stepping back there. Okay, the mitochondria is an energy organelle. This is what makes the energy molecule for the cells, which is called ATP. Okay, one of the misnomers, or one of the things that you often see when you're looking at mitochondria in cells is that they're very small, and some of the pictures that we use actually overstate their size. So don't get too wrapped up in our pictures. You aren't going to see a lot of mitochondria because they do tend to be relatively small. The mitochondria has two membranes, an outer membrane and inner membranes. The inner foldings are called cristae. And so we always talk about there being an inner mitochondrial matrix, the area inside the cristae, and an intermembrane space or an outer mitochondrial matrix, the area between the two membranes. So be aware that there's two different spaces. Those inner and outer spaces are very important in terms of how mitochondria work. And we'll go over in one of the later chapters all about how this mitochondria works. We'll spend a whole chapter on the production of ATP. And that's why at this point we want you to memorize what is the function of what is the function of the mitochondria. So the function of the mitochondria, it is the place where most ATP is made in cells by a process called aerobic cellular respiration. So aerobic means that it requires oxygen. Okay, the cellular respiration breaks down glucose in the presence of oxygen to make carbon dioxide and water, and also energy. Okay, the energy that's made in our mitochondria is made in terms of making ATP molecules. For every glucose we break down, we can make about 36 ATP. Now we're going to go back to another organelle that has two membranes that's real similar to the mitochondria, called the chloroplast. Take a look at this equation here. We're taking oxygen and glucose and making carbon dioxide and water, and in the process we're releasing energy. Let's look at what the photosynthesis involves. Photosynthesis is actually just the opposite. It takes carbon dioxide and water and uses them to make oxygen and glucose. Again, this glucose is the energy molecule that we eat when we need to make ATP. What drives this forward, again, if you go in the other direction, you release energy. This actually requires an energy input. And this little circle here is meant to represent that chloroplasts must absorb solar energy and use the sun's energy to drive this process forward. So chloroplasts actually work when light is present, and they make most of the energy molecules that autotrophs and heterotrophs are going to be used from there on out. So here's a neat picture of a chloroplast. Again, there's an outer membrane and an inner membrane. Within the inner membrane, you'll see these stacks of coin-like structures. We call those grana. Okay, they're very important in terms of the photosynthetic processes. This is where the chlorophyll is found. And then the spaces in between the stroma are very important. There's a lot of enzymes there. They're very important in terms of taking the energy that's captured and turning it into or using it in the processes that make energy molecules. Okay, there's other plastids that are filled with pigments, kind of like the chloroplasts, including chromoplasts and amyloplasts. Chromoplasts contain other pigments that have other colors, and they can also absorb energy and use that energy to provide energy for the cell. Amyloplasts, again, are granules that don't contain pigments, but amyloplasts contain amylose or starch, and these are starch granules can be found. Gosh, if you eat peas, if you eat potatoes, that kind of grainy feeling that you feel is actually this, when you bite in on those, that's actually the starch granules that hold the starch. Let's talk briefly about an interesting hypothesis. One of the things that I didn't talk to you much about with the mitochondria and the chloroplast is they actually have, along with those other structures, they have some of their own DNA, they have some of their own ribosomes. So a woman by the name of Lynn Margulis came up with something called the endosymbiotic theory of mitochondrial and chloroplast origin. And her theory says that basically she thinks that at one point mitochondria and chloroplasts were independent prokaryotic organisms that were taken in or to the cell by the process of endocytosis and came to live symbiotically within cells, and that was sort of the origin of those organelles. And it makes sense from a perspective. They've got their own DNA, they both have double membranes, and it does help to explain where they may have came from. Okay, again, here's a nice picture of that idea of where these double membrane organelles came from, and it would help to explain some of the evolution of why eukaryotic cells are more complex than prokaryotic cells. Okay. Now, the final thing I want to talk to you about, and again we've gone through a lot, is just sort of a theory of when you look at any one cell, you probably won't see everything that I I showed you, or everything that I discussed in terms of this introduction. What the organelles that cells have are related to their function. So what you find in a cell is going to depend specifically on what it does. Again, you can think about a muscle cell that has to move, it has to have lots of energy, it's going to have lots of mitochondria. Makes sense that potato cells have amyloplasts. Nerve cells have Golgi and vesicles for neurotransmitters, and that plant cell needs to have a vacuole, but none of my animal cells are going to necessarily have a vacuole. So again, you're going to find different organelles in different cells, and that's one of those reasons that the pictures that we show you when we show you a plant or animal cell are not necessarily representative of what you'll see when you actually go on and look at the histology of specific cell types. Alright, well that kind of ties this thing up. Again, the structure and function thing, those are key things that I want you to go through and make sure you have information on for each of the organelles. Good thing to do flashcards on. Every one of the organelles that was on these PowerPoint slides is probably going to be fair game for your exam. Thank you.